Alright, so we're going to start with Chapter 10 by looking at an overview of our muscle tissue. There are three main types of muscle: skeletal, cardiac, and smooth. Their primary function is to generate muscle tension. So generating force with muscle tension is the basic function of all muscle tissue. We have additional functions that they carry out, such as movement, maintaining posture, stabilizing joints. These are all going to be predominantly skeletal, the way we think of movement, but we also have movement within the body, movement of our blood, movement of the digestive system. All of that would be carried out by our smooth muscles. Generating heat, predominantly the skeletal muscle, and then regulating the flow of materials through hollow organs. Again, this would be via smooth muscle and cardiac muscle, because cardiac muscle is only found in the heart, and it is used to push blood around the heart. Here you can see how the appearance of the three different types of muscles vary. So cardiac and smooth are both striated, so they have these striations through them. They are both multinucleated, or skeletal's multinucleated. The cardiac looks that way, but it actually has a single nucleus. And then our smooth muscle is different in that it does not have striations. They are squamous. Remember back talking with epithelial, squamous means to be a flat, pancake-like structure. They have a single nucleus. Special features of our cardiac are that they have the intercartilated discs, which contain gap junctions so that the heart can rapidly move the ions from one cell to the next so that it can contract as a single unit. Also within those gap junctions are desmosomes, which are strong adhering proteins that help the heart stick together during the strong contractions that it undergoes. There is a table that gives you a summary of these, gives you the structure, the location, whether it is involuntary or voluntary. And so the only voluntary is our skeletal muscle, even though we don't really think about it. You learn to walk, so you have to teach yourself to walk, and then once you learn, it's still a voluntarily controlled thing. It's just you're not necessarily thinking about it as much as you had to when you were younger. And then the functions of the different muscles. And remember, when you're answering your possible exam questions, you need to give a little more detail than this to get full credit for that answer. So what are the properties of muscle cells? So this is muscle cells. All muscle cells have the property of contractility, which means that they have the ability to contract. They can shorten. They have excitability, so they respond to stimulus. They have conductivity, which means they conduct electrical charges across their plasma membrane. They can be extensibility, which means they can be stretched up to 3 times their resting length without rupturing. And because of this extensibility, it's important that they have elasticity, where they return to the original length after stretching. So these are our 5 properties of muscle cells. And so you have learned the generalized structure of a cell and the organelles that are within that cell. We have those same organelles within the muscle, but because muscle is a fiber, so it is a long cylindrical cell, a lot of the organelles have a similar but different name. And so your plasma membrane is now known as the sarcolemma. The cytoplasm is known as sarcoplasm. Myofibrils are a kind of like a cytoskeletal element, but they're the contractile elements, the contractile proteins within the muscle fibers. And muscle fibers are really big bags of these myofibrils. That's their predominant structure that you will see in there. Smooth endoplasmic reticulum is now called the sarcoplasmic reticulum, and you can see this blue net-like structure surrounding all the myofibrils. And remember back when we learned the smooth endoplasmic reticulum, some of you probably did not make note of it, but in the muscle cell, the big job of the sarcoplasmic reticulum is calcium storage. mitochondria. We have the mitochondria. We have lots of mitochondria, especially in our muscles that do a lot of work, long distance, not our short bursts. And we'll learn later in this chapter that the number of mitochondria, the myoglobin, so hemoglobin's in red blood cells, myoglobin is in our muscle cells, is what determines the activity of that muscle. And when we think of poultry, we think of dark meat, white meat. So the dark meat is the one with more myoglobin, the white meat is the one with less myoglobin. And then the nucleus, which is kind of pushed out to the outer edges of the cell because the myofibrils are contained all within. And so as I just mentioned, myofibrils predominant structure within the muscle fiber, 50 to 80% of the cell volume. They measure about 1 micrometer in diameter, so about the thickness of a human hair. They are specialized bundles made up of specialized proteins that allow for contraction. Other organelles, such as our mitochondria, again, as I pointed out, packed between the myofibrils. And then smooth muscles, myofibril arrangement is going to be different, as we'll see at the end of chapter 10. But cardiac and skeletal have the similar arrangement. That's why they're striated, and that's why our skeletal muscle, or our smooth muscle, is not striated. So how do those muscle fibers function? So here you can see the comparison of a muscle fiber in comparison to a red blood cell, a much larger cell. And you have to think like your thigh, your femur, the muscles that help your leg move. How long are those muscles running the full length of your thigh? So you can have some pretty long fibers running the length of your back. Also, you have muscles that run the full length of the back, and so those fibers are that long. And so our skeletal muscle consists of fibers, and they're surrounding the endomysium. So the endomysium is our innermost tissue, connective tissue, that we will find in our muscle fibers. Fibers can run as long as 30 centimeters compared to our red blood cell, which is only 7.5 micrometers. And so because most of us think more in inches than centimeters, 30 centimeters would be equivalent to about 11.8 inches, which is almost a foot, 12 inches. And so here is the basic makeup of a skeletal muscle fiber. And so you can see that the structure of a muscle is actually a bundle within a bundle within a bundle. And in an upcoming diagram, we'll look at these different bundles. But the muscle fiber itself is the innermost bundle. And it is made up of our myofibrils. So this whole cylindrical structure is our myofibril. Contained within the myofibrils are our myofilaments. And notice that anytime you're referring to muscle, we're using the prefix sarco or we're using the prefix myo. And so the myofilaments, we'll learn in just a few minutes, are going to be our actin and our myosin. The myofibrils are surrounded by the sarcolemma, which is the plasma membrane. And surrounding the sarcolemma is our outer connective tissue, the endomysium. Contained within the muscle fiber, we have the mitochondria, we have the nucleus, we have the smooth endoplasmic reticulum, but now it is called the sarcoplasmic reticulum. And you need to label it the sarcoplasmic reticulum, the sarcolemma, which is the correct terminology. There are special features with the sarcolemma. We have the terminal cisternae, and so where the sarcolemma also comes down into the muscle fiber itself, we have these tubes that invaginate down into the muscle cell, those are called the transverse tubules. And so the sarcolemma is the plasma membrane. It also delves down into the cell with invaginations called transverse tubules. On either side of the transverse tubule, the sarcoplasmic reticulum abuts right up against it, and those areas that butt right up against them are called the terminal cisternae of the sarcoplasmic reticulum. And so with one T-tubule and two terminal cisternae, we call this a triad. And this is important when we get into muscle contraction, and you'll see why it's important then. And then surrounding all that, we have the sarcoplasm, which fills the inner areas of the cell. And so our myofibrils, that are most abundant organelle, made up mostly of our contractile proteins, which are our actin and our myosin. As I mentioned earlier, that sarcoplasmic reticulum, the big job it has in muscle cells is to store calcium ions, which are also critical in muscle contraction. As I pointed out earlier, we have the transverse tubules, which are invaginations of the sarcolemma that delve down into the muscle cell to allow the action potential. So they're there for the action potential to be able to get down into the muscle fiber. And so they are continuous with that sarcolemma on the outside of the cell. And on either side, we have those terminal cisternae that make up the triad. So each of our myofibrils are made up of hundreds of thousands of our myofilaments. The two main myofilaments are going to be our actin and our myosin. But we have other types of proteins also contained within the myofibrils. So the actin and myosin you're going to see are our contractile proteins. They generate the tension. We have regulatory proteins. The two big regulatory proteins that we have are trypomyosin and troponin. And they dictate when a fiber may contract. And it's actually troponin, but I call it troponin because I can spell it. The structural proteins, these help maintain the proper myofilament alignment and fiber stability. And we'll see those with the elastic proteins. And so here are those proteins. We can see them up close and personal now. And so we have our thick filament, which is made up of our myosin. And the myosin itself is made up of a tail region, a hinge region, and a head region. And the place between the hinge and the head is the neck. We have a thin filament that is made up of the contractile protein actin. And so this is the actin subunit. And on the actin subunit, you see this actin active site. And so the actin active site is actually where the myosin heads are going to bind. And so here you can see how the actin surrounds the myosin, and then the myosin is going to reach out and grab the actin and pull the sarcomere. So this is a sarcomere closer together, and that's how muscle contraction actually happens. Surrounding the actin, so the actin's like a beaded necklace. You have your individual actin subunits, but the thin filaments are made up of these long chains of the actin subunit, and so that is the thin filament. But also consisting in the thin filament are two of our regulatory proteins that I just mentioned. Troponin are these three yellow dots, and then we have trypomyosin, which is this blue rope. And notice that right now that blue rope is covering those actin active sites. And then holding the myosin to, this is going to be the Z disc, you'll see that's another structural protein, are these elastic filaments. And so these spring-like filaments that we see here. And so this is our basic microscopic structure. And this will be in lab. So I will release a lab homework that you can practice labeling the microscopic anatomy of the muscle. But you need to know that microscopic anatomy so that you can talk about muscle contraction. All right, so here we see a blow up of that arrangement, and we can see these striations now that we typically see in a muscle cell. And so our actin and myosin are interwoven here. They overlap with each other within the muscle. And I mentioned this before, the sarcomere. So our sarcomere is our shortest, smallest unit of contraction. A sarcomere is delineated by the section of muscle that runs from Z disc to Z disc. And so this is a Z disc here. Attached directly to the Z disc is the actin, and then the myosin is attached to the Z disc via those elastic filaments. Within our sarcomere, we have an I band and an A band. And so light bands are where there are only thin filaments, so they both have an I. So I bands, light bands, I, only have the thin filaments. So you have an I in all of these. And with our dark bands are where both thin and thick filaments exist. And so our dark band, A band, so A band, dark, A. And so we have these alternating I bands, A bands, light bands, dark bands. And that is what gives skeletal and cardiac muscle their striations. Some other areas that we need to look at within a sarcomere. The M line is the middle line, M for middle, so is that middle area. And the area where there is no actin. So this H zone is the area where there is no actin. Notice with our I bands too, as I pointed out earlier, only actin. So H zone, no actin. I band, only actin. And then the A band is always going to be the length of the myosin. So the A band is the length of our thick filament, which is made up of our myosin. And so the A band doesn't care if there's any actin in there. It doesn't matter to it. But the I band does. So the I band's only actin. And so we'll see how this is important and how you'll use these zones and bands, and make sure you don't mix up zones, bands, discs, because those are all important. You have to label them correctly. And so here we can see the arrangement. And so where we have the overlap of the myosin and actin, you can see how 3D wise there is actin surrounding, multiple actin surrounding every myosin, and that every actin comes into contact with more than just one myosin. And so this is the 3D structure of the area that is our overlap. So this is the overlap zone. So outer edges of our A band. Now if we look at the H zone, which contains only myosin, that's all we're going to see is myosin. And if we look at the I band, which only contains actin, then we're only going to see these purplish dots, which are the actin. So it is in that area of overlap that we see how they intermingle. And so you can put your two hands together and slide your fingers across each other, and so they are overlapping. And we'll see a depiction of that in just a minute. And so here is our bundle within bundle and with bundle. So our myofibril, surrounded by our endomysium, is bundled into a structure called a fascicle. The fascicle is surrounded by the paramysium. And then all of our fascicles are bundled into our muscle by the epimysium. All of these connective tissues come together to form the tendon, and the tendon is what holds muscle to bone. Ligaments hold bone to bone. And so again, we have the myofibril surrounded by the endomysium. All the myofibrils are bound into a fascicle, which is surrounded by the paramysium, and then all the fascicles are bundled into a muscle surrounded by our epimysium. And then here's our big picture of everything that we've talked about. So our bundle within a bundle within a bundle, pulling out our muscle fiber, looking at an individual myofibril, and the overlapping of our thick and thin filaments in the sarcomere where muscle contraction happens. And so there are some disorders that affect muscle tissue. Duchenne muscular dystrophy is the most common one. It affects mainly males because it is a defective gene on the X chromosome. So you need 2 X chromosomes to have this disease. And it is because of a protein dystrophin, which is a structural protein that you find in our striated muscle fibers, that is abnormal. And so it breaks down, replaced by fatty or fibrous connective tissue, which causes weakening of the muscles. So the symptoms usually start to appear between 2 and 12, getting weakness and a waddling gait, and generally wheel-bound by the age of 12 and dead usually from respiratory or cardiac failure by the age of 20, because remember, cardiac muscle is also a striated muscle. And here you can see the difference between normal muscle tissue versus a Duchenne muscular dystrophy patient. And you can see all these deposits where the fat is replacing the muscle tissue. And here is that depiction of our sliding filament model, how the thick and thin filaments slide over each other. Remember, a sarcomere is our shortest contractile unit, delineated by the Z disc. So your palms are the Z disc, your fingers are the thin and thick filaments. And if you run them between each other, you'll see how the sarcomere with the sliding filament model is going to work. And when we do have that overlap in that sliding filament, notice that it is the I bands that get smaller and the H zone that gets smaller because the overlap is greater. So here is a relaxed muscle. You have some overlap, but not a lot of overlap. But then as the muscle contracts, pulls those D discs closer together, you have more overlap. And as you have more overlap, the I band, which is only actin, shrinks because more of the actin is overlapping the myosin. And the H zone, which is only myosin, shrinks because the actin and myosin are overlapping more. But what never changes, as I pointed out, is the A band, because the A band doesn't care if there's overlap. All it cares is if there's myosin in there. And the length of our myofilaments is not changing. So these actin and myosin, they are not changing. Their lengths are not changing. It is percentage of overlap that is changing when the muscle contracts. And they are all being pulled towards that middle area of the M line. All right, so that is our muscle structure and beginning to learn the terminology so that we can talk about contraction. Have a wonderful day. Let me know if you have any questions.